nucleic acid. Here the analysis of the nucleus showed out that the same nucleus has two types of nucleic acid. One is DNA and another one is RNA. Actually, the presence of nucleic acid was showed by Frederick Michel. In Pasteur in 1869, and he called to these molecules as nuclein. This nucleic acid or nuclein, that one is here studied very well in the 20th century. In the last century, in the 19th century, Frederick Michel identified nucleic acid. Laterally, that were found out in the acidic in nature. That's why it is called as nucleic acid instead of nuclein. And it is observed that after undergoing the analysis of nucleus, there are two types of nucleic acids are there. One is DNA and another one is RNA. Here these nucleic acids, these are further made of nucleotides. These are basic building blocks, nucleotides. These are the monomer molecule, similar molecule, the basic building block molecule. These building blocks here are nucleotides. Nucleotides are of further on the ground of its presence are of two types. The DNA are made of deoxyribonucleotides, whereas RNA are made of ribonucleotides. What is the difference between these two deoxyribonucleotides and the ribonucleotides? Both are, these are nucleotides. Each nucleotide is made up of three molecules. Molecule 1 is sugar, that one is ribose. Ribose sugar, that sugar is a pentose sugar. This is a monosaccharide sugar made of C5H10O5. This is ribose sugar. Second one is the nitrogen base. And the third one is phosphoric acid. Such three molecules clubs to form the nucleotide. Such nucleotides, one after the other, forms the chain of the nucleic acid. How these nucleotides appears or looks like, I will show you here. Nucleotide is with the ribose sugar. This ribose sugar is pentose sugar, and at carbon number 1, 2, 3, 4 and at carbon number 5, here at carbon number 1 always nitrogen base is attached. Nitrogen base, sugar. S stands for sugar, N stands for nitrogen base. Sugar is here 1, nitrogen base is 2, phosphoric acid is 3. Sugar and nitrogen base when combines, that forms a structure. That structure is called as nucleoside. Nucleoside. To this nucleoside, when a phosphate molecule H3PO4 is joined, then this becomes nucleotide phosphate or nucleotide. Nucleotide. Here this sugar molecule is, just now I have mentioned, is ribose, and the empirical formula of ribose sugar is C5H10O5. From this ribose sugar, when one molecule of oxygen when removed out, C5H10O4, oxygen molecule, when one oxygen is removed out from this oxygen, the oxygen molecule becomes 4 in this sugar. Then here to this sugar, when oxygen is removed out, that removed, for this removal word we are using D, deleted. What delete? Oxygen is deleted. From whom? From the ribose sugar. From the ribose sugar. From ribose sugar, oxygen is deleted. That's why it is called as deoxyribose sugar. This is ribose sugar. Ribose sugar is present in RNA. Deoxyribose sugar present in DNA. Such a separate sugar now gets formed here. In earlier case we have mentioned a pentose sugar is there. This is a monosaccharide. From this monosaccharide, here from the oxygen, one oxygen when removed out, here there is a deoxyribose sugar formation takes place, and this forms DNA nucleotide. Nucleotide appears like this. This is sugar. Two types of sugar are there. One is ribose and the other one is deoxyribose. Likewise, these nitrogen bases are further of two types. One is called as the purine type and the other one is called as pyrimidine type. Purine and pyrimidine. Nitrogen is of purine and pyrimidine. The difference between these two is pyrimidine molecules are small in size. One ringed structure, whereas these are big molecules, big in size and with two ringed structure. They are with two rings. This is what we may call purine molecules, and pyrimidine is with the single ring. This is what? With two rings and a single ring, purine and pyrimidine. These purine molecules are further of two types. This is adenine and the guanine. Adenine and guanine are the two types of purine molecules. Pyrimidines are of further three types. One is here the cytosine, thymine and the uracil. Uracil is a pyrimidine. That uracil is found absent in DNA molecule, and thymine is present in DNA. The place of thymine is taken out by the uracil in RNA molecule. RNA. Here in RNA, uracil is present only in RNA. Such four different types of nitrogen bases. Sorry, made mistake. Two types of nitrogen bases are there. Each one is further off here. Two types. But pyrimidine is in addition, uracil is present. That one is only present in the RNA molecule. Thus, 1, 2, 3, 4 and 5 different types of nitrogen bases could get formed. With this, when this nitrogen base, say for example adenine is here, adenine, this nitrogen base is joining with the deoxyribose sugar, then it forms deoxyribose sugar plus adenine. This is a nucleoside. This is now called as adenosine nucleoside. And to this nucleoside, when a phosphate gets attached, it becomes a nucleotide called as adenosine. Adenosine mono stands for one phosphate, adenosine monophosphate. That one is mentioned AMP. This is a nucleotide. This nucleotide formation takes place. With this, we will note here the Watson and Crick studied DNA molecule on the ground of X-ray diffraction technique and proposed a model. We are calling the DNA model by Watson and Crick. That model was proposed in 1953 and they received, along with Wilkinson, in 1962, they received a Nobel. The DNA X-ray diffraction were provided by the Wilkins and the model were prepared by the Watson and Crick. In this model of DNA, I will draw a picture of a little picture here. DNA is a double-stranded molecule, whereas RNA is a single-stranded molecule. Such types of nucleotides here, one after the other, they joins, and the big molecule, macromolecule, the nucleic acid molecules gets formed. DNA, RNA. DNA is double-stranded molecule. Double-stranded molecule. Whereas RNA is single. We will see little later. This double-stranded molecule here has a backbone of sugar and the phosphate molecule, and that one is running in 3' end to 5' end, whereas the other strand is there 1 and 2, thus it is called as double-stranded with two strands. Instead of falling like this, two strands are here, strand 1 and strand 2, two strands. Both of the strands are anti-parallel. This one runs in 3' end to 5' end. This strand is made of sugar and phosphate. This strand is also made of sugar and phosphate. And in between these two, here the nitrogen bases are present. These nitrogen bases are here always purine pairs with pyrimidine type. Pyrimidine. Purine is adenine. Pyrimidine is thymine. Thymine and adenine, between these two, purine and pyrimidine, there is always weak hydrogen bond is there, and there are two weak hydrogen bonds are there. Likewise, guanine purine is paired always with cytosine, and that pairing is occurred with three weak hydrogen bonds. Such a type of weak hydrogen bonding bonds the nitrogen bases inside to provide ladder-like structure, and because of presence of purine and pyrimidine ratio as per Chargaff, that one is always 1:1. One purine means whatever the amount of purine is present, the same amount of pyrimidine is present in the portion of the DNA molecule. Here this strand is in this fashion. Now, this little rest points you will come to know in gene, its structure in class 12. Now, RNA is there. RNA is single-stranded. RNA is single-stranded. This structure is with only one strand. But that one strand may get folded here and there, and they forms the complementary base pairing. They form complementary base pairing wherever the folding occurs. This is actually a single-stranded molecule. This is double-stranded molecule. But this is single-stranded molecules undergo the coiling, and coiling is around its own axis, and that's why it forms to be folded here and there. And in folding area, they show the complementary base pair. Here, this one, RNA is of two types. RNA is of two types. Type 1 is called as genetic RNA. Genetic RNA. And type 2 is non-genetic RNA. Non-genetic RNA. The meaning between these two is they controls the cellular activity. Genetic RNA controls cellular activity. Normally, this type of RNA is present in the viruses. You know, viruses are neither living nor dead. These are creatures, structures, crystalline in nature. Some viruses, especially in case of plants and in some animal viruses and some animal viruses. In these, the RNA is a genetic material that controls the cellular activity. But wherever DNA is present as a genetic material, there only here the non-genetic RNA are there, and these are of three types. Non-genetic doesn't involve in genetical activity. Here these are mRNA, other are rRNA, and third one is tRNA. Such three different types of RNA are there. Genetic RNA and non-genetic RNA are the two types, no doubt. Genetic RNA has no further types, but non-genetic RNA are further of three types, and these non-genetic RNA participate their role in protein synthesis. They play a role in this activity. All these three different types of RNA are synthesized. DNA synthesizes the RNA. Different types of RNA. on the strand of the DNA, on the strand of DNA. Here mRNA, that one is, here the prefix terms are there, M, R and T. This is the second letter of the alphabet. M stands for message. This RNA carries message. That's why it is called as messenger RNA. Messenger RNA. They carries message. Message in the form of triplet codon from the DNA for the protein synthesis inside the cytoplasm. This is straight one. And the account, mRNA account, total 5% of the total non-genetic RNA. 5% of the 100%. rRNA, these are present in ribosomes, ribosomal RNA. This RNA is occupying area, or these are present in the ribosomes. That's why it is called as ribosomal RNA. That counts here 80% out of 100% of non-genetic RNA. These are present in cytoplasm. All are synthesized in nucleus on their DNA and later transported into the cytoplasm. rRNA is ribosomal RNA, accounting 80% of the total non-genetic RNA and involves in here. This becomes the site of the protein synthesis. This is the site. On that area, the protein synthesis could take place. This is one more type of RNA is there. This is called as tRNA or sRNA. S stands for soluble. No one can separate them by the centrifugation technique. But the term T, but the letter alphabet T is utilized here, involves in transporting. Transporting what? They transport amino acid. Which amino acid? Activated. Activated amino acids are transported. To the site. Transported to the site of the protein synthesis. Here these appears, they are actually the smallest type of RNA. These are smallest type of RNA, and accounting here, the count is mentioned. This is 15% of the 100% of total non-genetic RNA. 5%, 15%, 20 and 80%, 100% non-genetic RNA. These are found in 15%, but these are very smallest in size. Smallest type of RNA, and that smallest type of RNA exists. rRNA exists in two form or two models. Exists in two models. One is hairpin model, and the hairpin model and clover leaf model. Hairpin model was proposed by Hoagland and others, whereas the clover leaf model was proposed by Hoagland and the others, and clover leaf model was proposed by, I failed to remember. Okay, this is what the DNA and the RNA molecule, the nucleic acid molecule in the class 11th. Okay, thank you.